So today I'm going to speak to you about 4 topics, each one of which I know less about than the previous: development, plasticity, learning, and memory. And it's good to give a lecture like this. Typically you learn from giving it. But I actually ended up learning less about development, plasticity, learning, and memory than I had planned on, because really I wanted to link this lecture as much as I could to Christoph's lecture last week on complexity, computation, and information. But I still want to give at least a running start at dealing with all of these issues to begin with. And I'm going to be discussing several very highlights of neuroscience over the past 70 years, beginning with developmental plasticity, in particular developmental plasticity of the visual system, and in particular Hubel and Wiesel's work on ocular dominance. And this picture here is probably the classic data slide, the classic anatomical slide that people learn in an introductory course on development or neuroscience in general, which shows how if you deprive vision to one eye, the representation of the two eyes on the visual cortex becomes very asymmetrical because of developmental plasticity. And I'll walk you through this slide in 5 minutes or 10 minutes or so. The second topic I'll deal with, again briefly, is synaptic plasticity. And this is the classic picture that one sees in any elementary neuroscience textbook on the phenomenon of long-term plasticity of synaptic transmission in the hippocampus. And again, I'll describe this slide later on. And then there's learning and memory. Again, topics that I know less about, but really in terms of fundamental neuroscience mechanism, ones that we know much less about than certainly synaptic plasticity. We know a huge amount of synaptic plasticity building on the work, on long-term plasticity, but all sorts of different types of synaptic plasticity. But I'm going to start the lecture with kind of a downer from the early phase of the neuroscience of learning and memory, and that's sort of the classic paper by Carl Lashley, who studied learning and memory and its neural substrate. And he used a word that was coined at the beginning of the last century, the engram, and he really popularized this word. And the engram is the memory trace, the physical thing that's left behind in a brain when an animal learns something. And really from Lashley down to the present day, it is one of probably the major holy grails that people are searching for, is the neural trace to learning and memory. And we have some foothold in this topic, really better from invertebrate work, the work most famously of Eric Kandel in the Plesia, and we have a good deal of knowledge of synaptic plasticity and how it might, but really might, relate to learning and memory. But Carl Lashley, from I think the late 20s, early 30s, all the way through the early 50s, was searching for the engram, the trace of memory. And unfortunately, because of for him, but he's started the field, the neuroscience of learning and memory, but the tools that he had at his disposal were very blunt. And he had 2 tools. One was maze learning, or other types of easily proceduralized learning, and this was predominantly in rodents, and the other was lesion studies that he performed both in rodents and in primates. And he wrote a famous book in the 30s, but really summed up his career in this paper in 1950, In Search of the Engram. And the summary is a downer. This series of experiments has yielded a good bit of information about what and where the memory trace is not. It has discovered nothing directly of the real nature of the engram. I sometimes feel, in reviewing the evidence on the localization of the memory trace, that the necessary conclusion is that learning just is not possible. You know? And he was tremendously influential, but I think he just was stuck in his time and place, and perhaps didn't make the best choice in terms of learning protocol. I think there's a increasingly well-known, but even back then there was a good deal of hints from the neurological literature that if you lose certain parts of your brain, most famously and most early, the brain centers for language, you know, very localized part of the brain, you lose the memory or the procedural act of speaking and understanding language. But for maze learning, what he did up top was, this is a summary of all the different cuts and aspirations that he put in a rodent brain, and on the left is really the summary, a very famous slide, where on the x-axis is the percentage of the brain that was lesioned, on the y-axis are errors in maze learning, and he found this very lawful, curve that he fit, that the more brain that was lesioned, the more errors in maze learning, and the only thing he could really conclude is that the memory for maze learning, which probably is correct, the memory for maze learning is a highly distributed memory. So what matters was not the area, but how much memory is distributed, and most importantly, the summary of Lashley is he found no localized engram. Here's a less poignant story from the work of Hubel and Wiesel. And starting really in the early 60s, very soon after they did their receptive field work in the cat and then the primate visual cortex, they began the series of experiments that really won them the Nobel Prize, the series of experiments on developmental plasticity in the visual system. And they found that if one eye was closed during the right time in development, which they called the critical period, that profoundly influences the cortical representation of vision through that eye, even when that eye is opened. And they had a number of physiological studies which showed, again, that if the eye was closed, or even if vision was affected by having the eye look in the wrong direction, the cortical representation of information from that eye was very degraded. And this was a lovely paper by Simon LeVay in their lab, who demonstrated by injecting a radioactive tracer in one eye and then looking downstream from, this tracer was transported from the eye to the thalamus all the way to the cortex, and when developed in the proper way, they could demonstrate the anatomical connection from that eye disynaptically into the cortex. And in a normal animal that had no deprivation, if they injected this radioactive tracer into one eye and look back in the cortex, the tracer shows up as white on these pictures, and you can see that the black and white stripes on the left are perfectly equal, that the representation from the two eyes are perfectly equal. If instead they deprived vision of this animal for a very long time, from I think it was several weeks for over a year, they deprived vision from one eye, injected the tracer in the non-deprived eye, and now you see that the white region, the region that receives anatomical input from disynaptically from that good eye, it's much more than half of the cortical area. It's the vast majority of the cortical area with just very, very sparse thin lines where there was no input from the other eye. You know, at one level, one could call this an engram. This is an anatomical correlate of plasticity, and if one wants to make the large jump that all forms of plasticity at some level are similar, then this is certainly the anatomical trace of a fairly radical plasticity study. So I wanted to go from these two early studies to really consider the general question of information that might be encoded or might be present in a cortical network. And I'm just taking a slide from one of my earlier talks. I think this was on the connectome talk. And just reminding you of the fact that excitatory neurons in cortical networks are the most numerous. They make thousands of connections within the local network, and also they, in most cases, project elsewhere. But today I'll be talking about the local network. But they connect only with 10% of their potential targets if we're talking about cortical inputs to, excitatory inputs to other local excitatory neurons. And that's a very well-known fact from a number of different types of information, predominantly physiological studies of recordings and slices. And I want to go beyond that, just simple observation to make a point that others have made, certainly, is that sparseness allows connections to be selective, and therefore selective connectivity is a wonderful substrate for the storage of information. So there are really two sides of the same coin, that certainly the entire brain can't be wired up randomly. If it were wired up entirely randomly, then it really wouldn't perform all of the magical computations that we know it performs in being selective for different orientations, all the way through being selective for faces. If it were completely randomly wired up, that wouldn't be true. And that's the idea of plasticity, and in the case of faces, learning that embodies a physiological attribute to make the neurons do something. But that really also is learning that involves memory formation. It's if we remember a face, it's really, I think, because we have neurons that respond to that face, and therefore the memory of that face is in the connections that lead into the neurons that see faces. That's one person's view, but I think it's hard to argue with if you put it that carefully. This is my next to last lecture, and I kind of wish I had started with this, but it's a perfectly good place to talk about three books that have been tremendously influential to me in chronological order in terms of when I read them, but I guess when they were written as well. Christoph last week spoke about David Marr, who was tremendously influential in the field of computational neuroscience and really started the MIT school, which really has, Marr's way of thinking, I think, has dominated much of computational neuroscience. And in all of his work, but particularly in his book on vision that was published in 1982, just the year after he died at a very young age, he spoke of his three levels, and Christoph spoke about this last week. The three levels of neural computation are the computational level at really what is being computed. And for instance, an edge might be computed, a face might be computed. That's sort of the what of the computation. And then there are two how levels. The first one is an abstract how level. What is the algorithm that the system might employ to create neurons that are responsive to faces or to giraffes, as you saw last week, or animals in general. And the algorithmic level, the example that Christoph gave last week was Reid's and Huber's and Poggio's Hmax model, which is a sort of a hierarchical model, each step of which is similar to the simple complex model of Hubel and Wiesel. And then finally, there's the physical level of how, what is the hardware and what features of the hardware allow the system to compute the algorithm in order to solve the computational problem. And this is actually the first book that I read in neuroscience. I studied math and physics, and the summer before deciding what to do, someone told me to read this book, and I'm glad I did. The next book is a book that I, and many of us, read in graduate school in the 90s. And it's a book called Quartic Onyx, which the name is what it is, but it's really, the subtitle tells the story, Neural Circuits in the Cerebral Cortex. And it starts with a very basic but nicely pared down discussion of basic anatomy of cortical circuits and basic physiology of cortical circuits. But the heart of the book is deals with signal propagation through neural networks, sort of dynamical theory, and how the biophysics and the connectivity of cortical networks underlie signal propagation. And the big point in this book is an analysis of dynamics and in particular synchrony, and how synchronous activity can be propagated through multiple stages in a cortical network or even within a local network in order to encode information. And sort of the key word that people think of when they think of Obelisc's work is the syn-fire chain. Patterns of neurons that, a sparse population of neurons that might fire synchronously, and that synchronous activity is propagated through that network in different constellations of synchronously firing neurons. Then the final book is one that I just picked up sort of late. I really I'd known about this book that was written really at the same time as Obelisc's book, by another one of Christoph's doctoral advisors, Valentinu Breitenberg, who died last year, and Almut Schutz, who is an anatomist who worked with Breitenberg. And again, in this case, the title says it all: Cortex, Statistics and Geometry of Neuronal Connectivity. And it's basically an anatomy book written by someone who became a great anatomist, and certainly with his colleague, a great anatomist, Almut Schutz. But he's primarily a theorist, and he wrote about the features of flesh and blood, of actual cortical neurons, and how the geometry affects connectivity and how connectivity affects computation. People hear a lot nowadays of something that's called Peters' rule, after Alan Peters, but it's a very strong version of something that Alan Peters might have said, but it's the title of a chapter, of sort of the key chapter in this book by Breitenberg and Schutz. And Peters' rule is the idea that the overlap of presynaptic terminals, the axons, and postsynaptic elements, the dendrites of two neurons, so that it's just the spatial overlap of axons with dendrites that gives the probability of connections, and there's no specificity beyond that. And of course, we know that's not true, certainly at the level of cell types, and in fact, in the chapter, it's called Peters' Rule and White's Exception, where Ed White is an anatomist who showed one type of exception, but there are numerous exceptions to it that have to do with cell types. but certainly we here and many people think that even within cell types, there's a great deal of specificity in cortical connectivity. So here's another slide that I showed earlier on. Just to give the idea that a wiring diagram can be represented as a matrix, and here I have 10 to 20%, but let's call it 10 to 20% of excitatory cells are connected, and the question is which excitatory cells are connected. And here, if Peter's rule were true, the network of, in particular, of one cell type connecting to itself, that matrix would be formally random. So we have on the x-axis, so on the left, it's just a spatial diagram of an assembly of neurons sitting in 3 dimensions with the blue lines connecting each point, and that's the wiring diagram of those neurons connecting with each other. It's pretty difficult to see that, so the easiest way of showing a network diagram is with a matrix, with the x-axis being postsynaptic neurons, the y-axis being presynaptic neurons, and let's say there are 100 rows, 100 columns, and a blue dot means that presynaptic neuron on row 17 connects with a postsynaptic neuron on column 62. And typically this is called the weight matrix, or sometimes the adjacency matrix, IJ. And if there's a 1 or some number which gives the weight, that's the connections. And we know, again, that 80–90% of these entries in such a matrix are 0. And then the question is there any structure in the matrix? And remember this random matrix, because it really, it'll come back at the end of my lecture when I begin talking about information theory. And if it's totally random, it has very high entropy, which is another measure of the complexity or randomness of a system. But it's possible, however, that it's not totally random. And these two pictures, I should have said, are from a really lovely review article by Helmstetter, Brigman, and Denk, who are Winfried Denk's group, who have been one of the real pioneers in the connectomics movement. And the simplest kind of structure that one could have in a network is just cell assemblies, cells that happen to be highly connected to each other. The blue cells connect to the blue cells, the red cells connect to the red cells, but there are very few connections between red and blue cells, which are the diagonals with just a very few number of dots. And actually on the left, this is one example where you can even just see if you have the physical connectivity diagram in 3 dimensions, you can see there are a bunch of red lines connecting red points and a bunch of blue lines connecting blue points. So what is a cell assembly? And this is one of the important issues in neuroscience, and certainly in more theoretically minded neuroscience. And again, for the simplest version, cell assemblies are cells that are hardwired together. Certainly it's not the only kind of cell assemblies. There are all sorts of ideas of transient cell assemblies, and then the classic example of a transient cell assembly, which really isn't manifested in the wiring diagram, is Obelis's SynFire chain. So it's an important, so the SynFire chain is a dynamical cell assembly. But I'm going to be talking about cell assemblies that are manifested in the network clearly, and again, I said the connectivity manifestation is these cells of different types are connected to each other, but not to other types. And that poses the question, what is it about these different assemblies that make them wired together? And if you've been coming to this series of lectures, you certainly can imagine that I would think that these cells that are connected to each other shares some receptive field attributes, so they could have similar function. But a simpler thing that would create a cell assembly is just simply cells that fire together, synchronous activity. If the blue cells fire together, the red cells fire together, then you could imagine, naively, you could imagine that they might be connected in some way, and that naive assumption is one of the classic rules of neuronal networks from a 1949 book, which has been tremendously influential. Donald Hebb's organization of behavior. Really, he actually worked with Lashley, both on human neurological issues, but also in primate work. And this book really is about the relationship between the physical brain and behavior, but he made a particularly clear statement, really a prediction of something that might underlie learning. And I might as well read it. Let us assume the persistence or repetition of reverberatory activity or trace. You know, certainly memory trace is the idea that it tends to induce lasting cellular changes that add to its stability. When axon of cell A is near enough to cell B, so if an axon is in the neighborhood of a dendrite and repeatedly or persistently takes part in firing it. And it's not just that they're coactive, but A actually influences B and makes it fire. And that also implies a temporal relationship, A before B. I won't talk about spike timing to plant-in plasticity, but it's, he both had the idea of co-activation, but it's sort of temporal activation as well. He predicted that some growth process or metabolic change take place in one or both cells. He considered both pre- or postsynaptic mechanisms of this plasticity, such that A's efficiency as one of the cells firing B, is increased. So he had it all in one statement, and really it was a tremendously influential statement, so much so that it became Hebb's law. It used to be called Hebb's rule, is what it's usually called nowadays. And Carla Schatz has a mnemonic for it: cells that fire together, wire together. So again, let's go back to what I showed you very briefly at the beginning. Because we're putting this on the web, I try to get as many things from Wikipedia. This is a Wikipedia version of, I guess, Bliss and Loma's experiment, late 70s, early 80s. And it's the simplest form, the simplest protocol for creating what they termed long-term plasticity. And I just repeated Hebb's words here. But the basic idea of this protocol is that there's a stimulating electrode in a fiber tract right here, stimulating electrode, and that when the axons are stimulated there, without even an intracellular recording, you can record extracellularly the synaptic potentials, essentially, that are evoked by exciting the fiber tract. And if one just takes a quiescent slice or an untrained slice, one can stimulate that fiber tract over and over again. And on the x-axis is minutes, and so they're very infrequent stimulations, every some number of seconds or so. And the y-axis is the EPSP field, the slope of that, the curve of the EPSP field. And it's actually, in a good preparation, that's a very, very static thing. The strength of the multiple synapses that are being excited are very, very stable and the same. Then a tetanus is given. The axons are stimulated very repeatedly at a very high rate, and this high rate presumably is very effective at driving the postsynaptic cells. And according to Hebb's rule, but really this is the first example of it, if those synapses become very, very effective for a given amount of time, then it would make sense, according to Hebb, that the synapses get stronger, And in fact, that's what happened. There's a very brief phase, which is short-term plasticity, PTP, post-tetanic potentiation, which had been well known. But the new phenomenon was long-term potentiation. Over the course of many minutes, the synapses are strengthened. So this is the first example of a long-term plastic change in the mammalian system that really followed Hebb's law, and it was taken as the prime candidate, and remains the prime candidate for middle term, hours, perhaps days learning. Primarily, it was in the hippocampus, which was for a long time has been implicated in memory. So in the last few slides, so that is my last really scientific findings on learning and memory. And in the remaining slides, I want to discuss the issue of how much information might be encoded in a network. And I think I'm about to make a statement which is not totally true and might offend some people in the field, but it's largely true, that there has been a huge amount, just tens of thousands of papers, I think is not inaccurate, on long-term plasticity, short and long-term, but in particular long-term plasticity of synapses in the nervous system. And there's plasticity of excitatory synapses, plasticity of inhibitory synapses. My wife, Carol Landesman, studies the plasticity of gap junctional synapses. So all the synapses in the mammalian CNS can be very plastic and have long-term plasticity. But there are very few examples where the actual plasticity of individual connections, or even the plasticity of a pathway, are absolutely strongly related to the learning of an animal. There are a few studies, one by Reid in the early 2000s, and a number of others where an animal learns something with a correctly dependent behavior, and the typical finding is that the attributes, the plasticity of neurons that are in the pathway that one postulates to be learning, those neurons are differentially plastic, but it's hardly an example of plasticity at this synapse underlying a learned behavior. So let me go to more speculative work, and I guess I'll have a bit of actual data, anatomical work, but I'm going to bring up the issue of structural versus functional plasticity in the cortex. This is one of 2 or 3 slides where I discuss a really lovely review by Mieciej Klosky, Bartlett Mell, and Karl Svoboda that appeared in Nature. And it's just a nice example of how one should differentiate different types of plasticity. It's fairly basic, but it's nice to keep them separate. One is weight change that, and this is the kind of plasticity that one talks about in synaptic plasticity, typically LTP, that we have a wiring diagram where the presynaptic neurons, that here they have an example with 100 potential presynaptic neurons and 10 connections onto a postsynaptic cell. And in a typical plasticity, synaptic plasticity study, the system starts out with synapse I being very strong onto neuron, postsynaptic neuron, and due to some kind of perturbation, and presumably some kind of perturbation that might happen in the intact animal, synapse 1 gets strengthened. But synapse 1 existed beforehand. And then there's a different type of plasticity where there's actually a physical wiring change. And there are a number of reasons why this strategy would be taken by nature to lay down long-term memories that if it's physically there and it wasn't there before, it's a way of really changing the structure of the network in a way that, until the synapse disappears, is much more stable, perhaps, than synaptic plasticity of existing synapses. So in this example, neuron 100 wasn't connected to the postsynaptic neuron, and something happens, and neuron 100 becomes connected, and the original neuron I that was connected is no longer connected. And this really brings one back to Breitenberg's book and Peter's rule. Breitenberg believed very strongly in Peter's rule, I think primarily because of the era in which he was writing, when synaptic plasticity, LTP, was the dominant mechanism for changing a network. And it certainly has theoretically has the capability of changing the activity of a network. There's an example that it existed phenomenologically in slices, and therefore, since there was no example of morphological plasticity when he was writing, then just by Occam's razor, he postulated that it was all weight changes rather than morphological changes that were important. But that really changed, I think, starting in the late 90s in the work of Charles Gilbert and Torsten Wiesel, and also Michael Merzenich. But really, the field became quite robust. The field of anatomical changes with learning or with plastic changes became robust with a lot more really clear experiments with, again, the advent of two-photon microscopy, when people became able to look into the brain of a living animal. And I've emphasized how two-photon microscopy is useful for calcium imaging to see the activity of neurons. But it's, in fact, an equally important aspect of two-photon microscopy is morphological imaging. And one would typically take a transgenically labeled neuron that expressed a fluorescent protein, image the dendrites, axons or dendrites, but in this case, the dendrites or the spines on a dendrite in the cortex, and just look at the individual synaptic spines that just amazingly one can go back to day after day. The same animal, the same dendrite, and the same spines on dendrites, and look to see if those spines appear and disappear, if connections can disappear and be formed. And Tobias Bonhoeffer's group has a number of nice examples. Karl Swoboda's group and others have a number of nice examples. I sort of split the difference in a review article by Karl Swoboda, and they reviewed a paper from Bonhoeffer and Huebner, first author Keck, where they revisited an experiment that Gilbert and Wiesel did in the late 80s, early 90s, where they made a retinal lesion. Rather than affecting the entire input from one eye, they just ablated a little bit of the retina, a little bit of the visual world, and that left a hole in the representation of the visual world in the cortex. And the question is, can the cortex react to that perturbation with physiological changes? And that was found in the 80s, 90s, with anatomical changes that Corinna Darien-Smith and others found in the late 90s. But here, in the modern era with two-photon microscopy, that Bonhoeffer's group found that there are plastic changes in the region that was just locally blinded. They found that spines turned over. So in the schematic that is in Swoboda's paper of Bonhoeffer's work, a laser was shined into the eye, a lesion was made. They counted the density of spines both in the lesion, which is red, and the unlesioned control eye, which is blue, and the number of spines didn't change. And the next panel is the real finding, that the x-axis is time after the lesion, 312, and then jumping all the way out to 63 days and beyond. And the y-axis is the survival fraction. In the control animal, a certain number of spines turned over, or in this case, disappeared, and roughly a third of the spines disappeared over the course of 10 weeks. But with the lesion, 90% of the spines turned over, and then in the final simplest graph, the number of persistent spines went way, way down the fraction of new spines that persisted. So these are brand new spines that were created. The first graph is disappearing spines. The second graph is new spines that stuck around for a week. 40% of the new spines stuck around for a week in the lesioned animal. So this is, another example. One could really call this the Engram, the physical trace of not a learned thing, but of an adaptation to a perturbation. So let's go one step beyond. Imagine what an Engram might look like in the second decade of this century, as opposed to in the 40s or the 90s or even 10 years ago. And it's a nice thing to go back to what Christoph showed last week, Poggio's and Riesenhuber's Hmax model, which, if you remember, had a hierarchy from, at the bottom, early vision, just primary visual cortex neurons, like simple cells that create the receptive field property of orientation selectivity to first approximation, and then the neurons that, to first approximation, abstract that quality and make it more general rather than encoding a visual a vertical bar right here, encoding vertical bars that might exist anywhere in a region of space. So abstracting the idea of vertical in the complex cells. And the nice thing about Poggio and Riesenhuber's model is that they make a multi-layered neural network that has some learning rules, but is constrained to have this this hierarchy, first of a simple cell-like pooling of presynaptic inputs that might have a common, or that might create a new feature. So really creating a new feature detector, and then the complex cell level, which abstracts or generalizes that new feature. And then it creates mid-level vision. And what I wanted to emphasize here is that early vision, most of us think, to first approximation, that early vision is created by developmental plasticity. That actually may not be true in all species, and there are hints, or even more than hints, that there's more plasticity in primary visual cortex or primary sensory cortex of rodents than there is, say, in primates. Mid-level vision, it's hard to say how much of that is early developmental and how much of that is later plasticity and learning to first approximation. But certainly high-level vision has to be learning at some level. If we have neurons in humans that respond to Jennifer Aniston's face, that has to be a learned attribute. And if one is working in the Hmax world, it's this high level of simple cells that put together the features that together make a human face on the left, or a Dalmatian on the right. It's that stage of plasticity that might create the representation of something entirely learned. So finally, I wanted to go back to a bit of the paper that I introduced earlier. Finally, I wanted to go back to information theory, which means I'll try to hurry a bit. What is information? It's closely related to entropy, a common measure of complexity. So I think the simplest thing is that it's very difficult, or one could prove, if one accepts the definitions, one can prove it's impossible to transmit, store, encode, decode information without a given base level of complexity of a system. And that was formulated by Shannon in the 1940s. at, I guess, MIT and then Bell Labs. If a system X, so this is just an abstract system X, can have states little x. So X can be a brain, X can be a computer memory, and the states X, or the configuration of a hard disk, say, each have probability P of X. Then the entropy of a system is, we have a few equations here, entropy, H of capital X being the system, is minus the sum over all X, X being all states, of the probability of X times log base 2 of the probability of X. What does that mean? That's kind of a hard one. Why would that be true? And I didn't have time to write it down, but let me give a very, very simple example. Consider the numbers from 1 to 256, and consider them all equally likely. Okay? So you have a 256-sided die, and you roll it. The probability of all of those numbers is the same. What's the probability of those numbers? 1 over 256. Okay? What's log 2 of 1 over 256? That's a hard one. But it's actually not so hard if one knows what logarithms do. Log of 1 over number is actually negative of log of that number. So log of 1 over 256 is minus log of 256. So that's where the funny minus sign comes from. It's just because the probabilities are fractions that add up to 1. So in the case of a 256-sided die, all of them are equally likely. All of them contribute log 2 of 256. Log base 2 of 256 is 8. 2 to the 8 is 256. All of these things are equal. All of them have probability 1 over 256. So all of those probabilities of the P of x's can come out, and you just have 1 times log of 256, or 8. So, and that's what information is. You need 8 bits to represent a number from 1 to 256. It's nothing more complicated than that. It's just a generalization of that. So the entropy of a hard disk is 8 times the number of bytes, period, assuming that random things are written. That's kind of boring. So really the important thing is mutual information. You have X, the system, that under some kind of randomness assumption has this entropy. And now we have a model of what entropy is. It's the information storage capacity in one way of thinking of it. The mutual information between two things, X and Y, is the entropy of system X, which I've already discussed, and then something complicated, the entropy of system X given Y. And the simplest thing to think of Y is the decoder. Y could be all sorts of things. It could be the message coming out, but the simplest thing of it is to think of it as the decoder. And now, what's the simplest decoder of 8 bits? The number. So mutual information between our magic decoder of 8 bits and the 8 bits themselves is the entropy of the 8 bits, 8, minus the entropy of those 8 bits given the number. So what's the entropy of these 8 bits, 1, 0, 0, 0, 1, 0, and 92? How much randomness is there between those two things? And the answer is exactly 0 entropy. There's no uncertainty. If you know the number, you know the bits, and vice versa. So the second term disappears. So the information, the decoding of these 8 bits, is simply the entropy of the encoding of the substrate minus what you can figure out about the substrate given the magic decoder. It's a little bit complicated, but I hope that gives an idea. So a hard disk has infinite entropy. It just has all of these zeros and ones. We know how to read it. So every one of those zeros and ones is potentially decodable by your computer. And that's what decoding is in a system we really understand and that doesn't waste any bits. So information theory is the study of information transmission. That was the original sort of use. Encoding and decoding sort of comes into that. But really storage and retrieval in the computer age, but really in the Engram age, storage and retrieval of memory is sort of the issue and how it relates to entropy. That's really the interesting quantity. So why is it interesting? If we want to read out a neural system, it's important to know how much information it can encode. I just have two more slides. One from a period in my life that ended 10 years ago, where we were looking at spike trains in the thalamus and the cortex a lot, and with two really wonderful postdocs, Pam Reinagel and Yang Dan, we did a number of studies on information in neural signals using what I told you about. And the bottom line of this study that we did 12 years ago is that the LGN can encode 3 to 4 bits per spike. And in this case, it was potentially encoded, but we and others have papers where we actually decoded this information. But this is an easy sort of exercise in what entropy, how it relates to information encoding. Up top, we have LGN responses to random stimuli. We gave a bunch of randomized stimuli and just looked at the LGN responses, and lo and behold, they looked very, very, very random. All sorts of different motifs could come out of the firing of an LGN spike train. So the LGN spike train has very high entropy. But is that used? Is that entropy used, or is it just pure noise? An easy way, we didn't invent this, other people, really largely influenced by Bill Bialik and the people he trained, had this way of looking at the mutual information between stimulus and response, is taking a repeated stimulus, a very strong repeated stimulus that did a good job of driving these LGN cells. And here are probably 100 or more repeats of the same stimulus. Here's the stimulus. And here are the spikes over time that the LGN cell responded. So very high entropy in general, very high reproducibility when the same stimulus is given. So mutual information is the entropy of the whole system minus the entropy of the system given some outside knowledge. So here it is again, the entropy of the response and the stimulus, the mutual information is the very high entropy of the response minus the very low entropy, the very high reproducibility of what we see in the response given the stimulus. So the LGN is a great, and the retina of course, great information channel, getting information from the outside to the inside. And it's sort of, at first, non-intuitive, that each spike can give more than one bit of information. It's not just yes or no, because it's not equally, spikes are kind of sparse. So a spike is a rare event, and if it happens rarely and very precisely, that can carry 3 or 4 bits per spike. Okay? And that's sort of, this is an aside, but I hope you see the analogy in the final slide. So how many bits can be stored in a synapse? So we really have a good handle, frankly, a great handle on how much information the activity of the brain can transmit about the outside world. We have almost no idea of how many bits are stored in a brain. We really don't. We know it has to be high because we can do amazing things, but we're at the same depressing place that Lashley was, frankly. We really don't know where these memories are. But this nice review by Kloski, Mel, and Svoboda give a hint of how much memory could be in a circuit, just in physical wiring. And they gave the example, if there are 100 neurons, each of which receives 10 connections, then the entropy per neuron is this, log base 2 of 100 choose 10. 100 choose 10 is a binomial coefficient, or if you remember Pascal's triangle, it's the 10th row of Pascal's triangle, 100th row of Pascal's triangle, 10 numbers in. It's the number of different ways you can choose 10 objects out of a hat with 100 labeled objects. And if you do the math, that gives you 46 bits for these 10 synapses. So in this simplified case, there are 4.6 bits per synapse. So a synapse is more than a bit. In a sparsely connected network, it's 4.5 bits. And that sort of generalizes. They give very simple arguments, but one can give simpler, complicated arguments for the general idea that if there are n neurons in a circuit and there's 10% connectivity, then there are about 4 bits per synapse. Okay? So how much of this information capacity is used, and can we decode it? I wanted to put a slide in, but ran out of time. In a cubic millimeter of cortex, there are 100,000 neurons and a billion synapses. So in all of visual cortex of the mouse, there are roughly 5 or 6 giga synapses, 4 bits per synapse. That's 2.5 gigabytes of potential storage in visual cortex. But the question is, can we read it out? So again, we have this thing, but only if we know the code. So we have this device that could store 2.5, 3 gigabytes of synaptic memory. Some people believe it's fairly randomly wired and therefore wouldn't store very much information. Some people, myself probably included, believe that there's a lot of specificity and therefore it uses a lot of this information capacity. But in order to understand this, we need to be able to read it out. The information, the mutual information of X, the synapses, are a couple of billion synapses, given Y, which is unknown, is the entropy of X, we know that, a couple of gigabytes, minus the degree of regularity between X and Y, between Y, our decoder, and X, our system. And if they're highly dependent, that number could be 0, and it's like a smart disk drive. It certainly isn't that, but we need to know how to read it out. So if we could relate the synapses to a learning paradigm, that might give us a tiny hint of the relationship. If we could relate the wiring diagram to receptive fields or correlated activity, that might give us more. It might give us more degrees of freedom to predict the wiring diagram from the physiology or vice versa, which can give us more information. And perhaps we could glean the most information if we could just look at the graph itself. If it's highly dependent upon itself, that reduces the entropy of the system. So this, nature has created something that's effectively infinitely complex. Our cortex has 1 trillion or so synapses. Are those, and the ability to encode, it's more than 1 trillion, but trillions of bytes of information. We're going to have a hard time finding a decoder for all of it, but if we have something like a connectome, something like the connections within a circuit, we might be able to say something that we assume is random and therefore has high entropy, in fact is very non-random in this case, and therefore that it has a lot of information stored. But really, we're nowhere near any of this. I mean, we're almost as far away as Lashley is, but I think a big part of what I'm interested in, and I think the Institute is interested in, is finding the why that can help us read out the physical or even just the synaptic strengths in a network to look for the information in the computer program, which is the input-output thing that creates receptive fields, or really, the flip side of the same thing, the memories, really, and I think that is the closest thing that we might get to an engram. So I'll stop there. The question is, it's the obvious question, given the only numbers I threw out there, that in the LGN and in the retina, we and others find that a typical number of bits per spike in information encoding is 3 or 4 bits per spike. And then from this theoretical paper by Schlafly, Mel, and Svoboda, they came up with the number that there might be 3 or 4 bits per spike, 3 or 4 bits per synapse in a network that has 10% connectivity. What's the relationship? And there is no relationship, really. That one is about information encoding in the periphery. The other is about the sparsely connected cortical network. We know tons about encoding the periphery, so you can talk about where that number might come from. We know nothing about what information might be encoded in a cortical network, so there's no relationship. Where did we get the number, the probability distribution? It's actually quite simple. Based on the work of Strong and Bialik, people who influenced this, what we did was we made words composed of, I think in this case, 8 time bins. And those words consist of number of spikes per bin. So typically there were 0, 0, 0, 1, 0, 1, 0, sparse strings of zeros and ones, rarely a 2. And if you look at all of those words, that object has a certain entropy. If we look at those words contingent, and that entropy of all those words was probably 5 or 6 bits per spike, just because of the sparseness of the spikes. It'd be 1 bit per spike if they were exactly half the time. It would be 8 bits per spike if there was a spike every 8 bins, and I'm sure that's wrong, but work with me. So essentially, it's the probability distribution of these words. It's the right question again. It's a generalized connectome question, and it's the right question. Connectomes viewed through the lens of information theory. The question is, given that every animal is different, yes, you can say that the denominator of the entropy of the system is very high. How do you find relationships if every animal is different, every stored memory is different? And the answer is, I think it's going to be extraordinarily hard. And I think the question, however, it can have the flavor, now I'm just thinking out loud, have the flavor of the physiology. If we can find, certainly we're not going to find neuron X in every animal has this constellation of inputs of 5,000 inputs, 1 through 5,000, voila. But certainly we don't find neuron X in every LGN cell that's looking here, that has this dynamics, that responds to this stimulus. It's more true in physiology, but still, every instant is different. I think it's the regularities that help us out. And I think if we could find the magic factor that allows us to predict the connectivity perfectly, then we could say the system is using all of the entropy. If the magic factor is the receptive field, we're not going to predict all of the connectivity. And again, I'm thinking out loud because I'm not sure the math allows this, but pretend that we don't have that. So pretend we can do that, and we can say, yes, there are lawfulnesses in the structure-function structure correlation that says something is being stored here. This is fascinating. Let's keep looking. Perhaps at the end of the day, if we have a full connectome and we take the network -1, and we can predict all of the inputs to that -1 from the network, that would be an example of a very strong mutual information between network -1 and full network, and that's a great reduction of entropy. I don't know if the cross terms will kill you. It's probably not going to tell you all of the encoded information, but it will tell you that there's a lot of encoded information. So it's really trying to find very, very strong dependencies between a measured thing and everything else.
For that, mathematically, do you need just one connectome, or do you actually need many?
I think you only need one connectome because it'd be good to do two, but it would be an extraordinarily strange world where in one connectome, and you wouldn't do it with just one, predicting the inputs and outputs from one neuron, you'd do it from all of the neurons in the network. What's the average predictability of the network with respect to neuron I, where I varies? If that's very, very lawful, it'd be pretty strange if that number varied in different animals, but actually, again, thinking out loud, that might be an amazing parameter to have in neural networks, that in the crystalline visual cortex, that parameter is highly predictable. A lot of the information is used in prefrontal cortex that needs to learn, or parietal cortex that need to learn, that might have less reliability because it needs to learn more. So, but again, just musing about a science that doesn't exist yet. So the question is, can these ideas be used to estimate the degree of distributivity of the network, local versus long distance? I don't know. I'm just making it up for local networks. I really, it doesn't exist. And then doing the extra, not epicycle, but extra degree of difficulty of local regularity and connectivity / computation versus global. You could think about it, but I wouldn't want to now. So Stefan always has 2 questions, even 3 questions, even if it's only one. Okay. 3 topics. Topic 1, that if you consider the local network, then the 10% connectivity rule works, and it's roughly 3 or 4 bits per synapse. Another part of the Shklovsky et al. Paper is that if it's a sparser network, and for sparser network, Reid distributed network, or in fact the entire brain, then P is not 10%, but P is 1%, 0.0001%, et cetera. And if that's the repertoire, then it can be hundreds, thousands, millions of bits per synapse. Sure, yeah, and that's sort of Mike's question. That's hard. Second part is also in the paper that they consider graded synapses, and they, they say if there are 4 states-ish, then that's 2 bits per graded synapse, even if it physically exists. Then the final question was something else, which I forget. And then if there are repeated patterns. Yeah. Yeah. So, and this they deal with. If there are repeated patterns, in a way, a repeated pattern can be either a way of encoding information, or it is potentially a way of subtracting the amount of information that you can encode. If always triples are connected very strongly, that might actually decrease the entropy. That does decrease the entropy of the whole system. And if that motif is a bug rather than a feature, then the entire entropy of the system is less, and less can be encoded. I see that glass as more half full, that the higher order, the existence of those motifs is a way of radically and simply decreasing the entropy, and therefore having more encoded information. But again, rank speculation. I think we should stop here. Good.